Good morning, everybody. Welcome to Turfgrass Epistemology. I'm Travis Shaddix. Here on this channel, we are on a never-ending journey to find out how we know what we know about turfgrass science. Thank you for coming today. I see Lush and Gray and Brady. You guys, Looney, everybody's already in the chat this morning. Welcome. So happy to have you all here. We had a good night last night with Dr. Raymer. That's what a leader looks like in the turfgrass industry. I mean, Paul Raymer is awesome. I'm really happy he came on. Had a good show last night. Today is another great scientist who worked with Dr. Raymer on these publications with thatch. We're continuing to go over thatch today. And we have next week, we're going to be going over thatch. And then next week, I swear, is the last week I'm doing thatch for a while because I am not comfortable with thatch. I don't know much about it. And it's definitely an area that I've learned about more going over these papers, really, than I ever did before. So next week's going to the last part of thatch, and we'll go on to some new topic. My guest today got his bachelor's degree from Punjab Agricultural University in India in 2005. He got a master's degree in 2007 from India, from the Punjab University as well. And then he went to the University of Georgia, as we mentioned last night, and got his PhD with Paul and Bob and the guys up in Georgia in 2012. And I believe now he's at the University of Florida. My guest today is Dr. Sudeep Sidhu. How are you, Dr. Sudeep? Did I say your last name right? Sidhu?
Sidhu, yes.
Sidhu.
Good morning. Yeah, good morning, Travis, and good morning, everyone. And I'm happy to be here.
Thanks so much for coming. For those who might not know, why don't you tell us a little bit about yourself, Sudeep, and how you got to where you're at?
Well, it's a very interesting and long story, but I'll start. I grew up in the state of Punjab, which is in the northern part of India. We are primarily an ag state. Area-wise, we are less than 2% of India, but we produce about 20% of India's wheat, 12% of India's cotton, and 11% of India's rice. Oh. So I grew up in a farming community. My dad or my uncles, I should say, while growing up, we had our own row crop farming operation, dairy farms, and poultry farms. So kind of grew up on a holistic ag, family operation, and so had a natural inclination towards agriculture. And moving forward, went to Punjab Agricultural University, which is one of the premier ag research institutes in India, and did my bachelor's and master's. And I was trained as a soil scientist up until my master's.
Okay.
And so Punjab Agricultural University at that time didn't have any turfgrass program. So my master's research was on soybean. And so I was in the fertility work, I did sulfur and boron studies on soybean and their impacts. And then it's just destiny that I ended up with great scientists such as Dr. Paul Raymer and Dr. Bob Carrow, Dr. Jack Huang, and got to work on this very unique research, which is the use of ligase enzyme on thatch. So I worked with Dr. Raymer on this project as part of my PhD for about 4 years. Okay. Then I worked with Dr. Patrick McCullough at UGA as a postdoc for 2 years, working on herbicide physiology. So did a lot of carbon-14 related stuff to understand the uptake, translocation, and metabolism of different plant growth regulators and herbicides used in turf grasses. After that, I got an opportunity to go to Nevada and work as an agronomist growing vegetables for a production company. Oh. So grew vegetables for about 3 years for them, and decided that I just like to be back in academia. And so I got an opportunity to work with Dr. David Wright at University of Florida, who was housed at North Florida Research and Education Center. So started working with Dr. David Wright as a postdoc, and started working on precision ag techniques to improve nitrogen use efficiency in cotton. And started working on some alternative crop rotation systems such as sod-based rotation, where we incorporate Bahia grass in our row crop land. And so so worked with Dr. Wright for 2 years as a postdoc, and then wrote a grant and had my own position in a grant as a non-tenure track faculty member. So stayed with Dr. Wright's program as a non-tenure track faculty member for 2 years, working similarly working in the similar kind of projects related to peanut, cotton, and corn, basically on nutrient management.
Okay.
After that, went to another location that's also part of North Florida Research and Education Center, but that's in Live Oak. So I worked there as 100% extension faculty. A regional specialized agent in agricultural water management was my title. So again, I was dealing with a lot of nitrogen in corn related extension programs and research programs. And then last year, Dr. David Wright, my former boss, retired and his position came open. So I applied for the position, and starting this September, now I joined back at NFREC in Quincy as an assistant professor of agronomy.
Oh, so you're a few months into your assistant professor position.
Yes.
Oh, okay.
Yes.
Well, congratulations on that. Good for you.
Well, thank you. Thank you. I always feel that sometimes you can't control when life takes you to different places and gives you the opportunity to work on different. I never thought that I would work on turfgrass.
Okay.
And then at the same, because I grew up in a farming community with row crops, growing wheat and rice. So never thought of working on turfgrass, and then at the same time, never thought of working on vegetables, but God has his own plans.
Yeah. When a door closes, a window opens, and you got to take advantage when the opportunity is there. Yeah. So you basically have a very similar story as a lot of my generation and the prior generation here, where we kind of grew up on farms, or at least in that it was in our sphere of influence when we were growing up. And we sort of gravitated towards that as an interest. I often wonder, what's the next generation going to look like? Because we just don't seem to have the same, input or interaction with plants and soils and farming as I did. Maybe I'm wrong, but it just doesn't seem like we have a lot of the students coming up nowadays who grew up in that environment.
I agree with you, and this is, I think this is just sad. And this is just not the situation here in the U.S., but it's the same back home. So I went to meet my dad last year, and just when we were talking as a family, me, my uncle, my dad, and everybody was sitting, and the general consensus was that my generation, the guys and the girls in my generation, in my village, they've left. Somebody's in New Delhi, somebody's in Bombay, Mumbai, and they're doing IT jobs, or most of the folks went to abroad, different countries abroad for their higher studies. Yeah. So there are just a handful of young boys in my generation left back in my village who are still farming or still somehow connected to farming. So it's just it's a sad situation.
Yeah, it's the next chapter in human civilization, I guess. And I'm curious, like, man, are we gonna be okay? I guess. Well, we'll figure it out. Well, then you ended up going to Georgia, like I said. And did you know the legendary status of the professors at Georgia before you joined that team? Did you know who they were?
Well, I would say partly yes and partly no, because it's a funny story how I ended up at Georgia. So I started my PhD program working at Oklahoma State University in the spring of 2008, working with Dr. Bill Ron, who at that time and still is one of the greatest, considering nitrogen work or optical sensors with nitrogen, work. So I started working with him, and my wife, at that time, my fiance, was working at University of Georgia. So it so happened that initially we decided that both of us it's fine for us to be at two different locations and finish our PhDs. But soon enough, we realized that no, that's difficult to be at two different places. So at that time, this opportunity came at University of Georgia Griffin campus, and I applied for and got the assistantship. So,
Okay,
but
good.
at that time when I was applying and I was looking for opportunities at University of Georgia, that's the time when I actually came to know about the huge status of Dr. Raymer and Dr. Bob Carrow. So that's why I said partly yes, partly no.
Yeah. Well, there's some people that just, you know, their success and their impact precedes them, and Bob and Paul, I mean, not to say anything about the other authors are great as well, but Bob and Paul have been around for so long, and their impact is just, it's going to be felt for many decades. They're very, very good at what they do. I hear Paul's going to be maybe setting off into the sunset here in the next, in the future sometime. I don't know, but we always, they leave this massive void when they leave. It's like, man, who's going to fill those shoes? So hopefully, we are able to fill those shoes. Well, today you're on here because I read one section of the paper we're going to go over, and I'm just like, I don't know what he's talking about. This is so far beyond my ability to comprehend. I need to get Sudeep in here and help explain it to me. And we've been going over thatch for about a month now, Sudeep, and we've talked about mechanical issues and how to culturally manage the thatch, via destructive methods, as I'm sure you're well aware of those. And then this idea in the 60s came around, like, can we do this without destroying the turf? Is there something we can do? And there's been hits and misses. They've tried sugars and they've tried enzymes and they've tried, all sorts of bacteria. They've tried all sorts of stuff, and very rarely does anything ever work. And if it does work, like nowadays, there's a little bit of hint and miss about some types of sugars, but those are, I think they're fairly weak. We're going to go over those papers next week. There's not a lot of strong evidence for that, but there's a little bit. And the same thing happened with lykase in 2011 and 12 when you published the Hort Science paper. It's like, well, there's a little bit here in this greenhouse study, but since then you've continued to put out papers in the field studies with different grasses and different settings, and we see similar responses. And so I wanted to wrap up, we've discussed this on my channel is where I'm going, and I wanted to wrap up this lykase topic we've been on for the last 2 or 3 episodes with the review paper you guys published in the International Turfgrass Society Research Journal, because it sums up sort of all the work you all have done. And there's a lot in this paper, Sudeep, as you know, you wrote it. So there's a lot in here. I want to get through here, but I would like to, question you specifically about this one or two paragraphs that describe lignin, and it describes to my audience and to me what exactly is it, and what exactly is lykase doing to help facilitate the degradation of the thatch, of the lignin in the thatch. So I'll get to that in a minute, but just briefly, do you want to just give us a bird's eye view of this particular paper? And I know it's an amalgamate, it's a conglomeration of all your projects, really all into one paper. You want to kind of just give us a background as we go into the paper?
Yes. Travis, this whole idea about using lykase to manage thatch started back in 2008. Mm-hmm. And I was the grad student, PhD student, working with Dr. Jack Huang, Dr. Paul Raymer, and Dr. Bob Carrow was on my committee at that time. So the whole idea was that how can we basically enhance organic matter degradation, particularly in the thatch layer. Now, as you mentioned, there have been several studies done in the past where sugars were used and different substrates were used or different inoculates were used. Some of the issues with using sugars per se, or using other inoculants or microbial populations is if we apply microbes directly on turf grass, right? For us to be able to grow a particular microbe that we actually intend to be there to produce enzymes that can degrade lignin, and I'm coming back to lignin, that's basically very difficult in intensively managed turf to create that microenvironment mm-hmm. for that particular microorganism, right? So if you are using sugars, as that sugars have been used in the past, right? So it's basically a temporary phase to improve microbial population. But it's not about the increase in the microbial population. It's also about sustaining that population over a longer period of time, right? That's the first thing, sustaining microbial population. Second, it's just not any microbe that we need. It's very particular microbes that can actually degrade lignin. That's what we require. So what happens is, and I'll try to be very generic here, and I'll use my hands to explain, why we thought of. Consider my finger. So consider my finger as a cellulose unit. In a plant cell wall, the primary unit is cellulose. Is the most abundant naturally occurring molecule is cellulose. And this is readily decomposed by bacteria. Most of the bacteria, most of the fungi can eat it up really fairly quickly. All right.
Okay.
But this is covered in a matrix of hemicellulose. All right. Sort of. Not always covered. Sometimes hemicellulose and cellulose strands are side by side, or they are just different strands in the cell wall. But both hemicellulose and cellulosic sugars are readily decomposable by most of the microbes that are present in soil. But the thing is that if you look at cellulose or hemicellulose strand, again, I'm using my finger, both these type of sugars are actually covered by lignin.
Okay.
All right?
Okay.
Now imagine that you have plenty of bacteria and fungal organisms that can eat up your cellulose, which is your major source of, about 35 to 40% of your cell wall is cellulose. But now that cellulose is not available because it's protected in the matrix of lignin, right? So the thought process was that we should not target cellulose. We should not target to somehow decrease cellulose, because that we knew that most of the microbes in the environment are capable of doing it. So the thought process was that the outer layer, this lignin matrix around cellulose, break that down. If we are able to break that lignin matrix or open up the bonds, then we will be able to, and this was just an assumption at that time, our theoretical assumption, that once the lignin is open, that lignin structure is broken down, then the microbes will have access to the sugars, which are cellulose and hemicellulosic sugars. So this is the baseline. This is the basic, thought process behind this study. Okay.
So since we're on that topic, and I appreciate the introduction, I want to, because you went right to the area that I want to ask about. And you have very good explanation. I like your use of visual usage of explanation of what it is. But you have to understand, I'm a soil scientist, but I know nothing about lignin. Everybody who's listening to this knows more about lignin than I do. So if I'm hearing you correctly, lignin is not a structural component. I'm going to repeat back, and you tell me what I'm wrong and help educate me here a little bit. Lignin is not necessarily a structural component. It's a coating on structural components that prevent degradation of those structures?
No.
Okay.
Lignin is a structural component.
It is, okay.
Of the cell wall, primarily of the secondary cell wall that we have.
Okay.
It is a structural component, but the way the components are in the cell wall is you have your sugars, but they are covered in a matrix of lignin.
Okay.
All right. And now let me clarify a little bit. Then what is the importance of lignin, right?
Yeah.
The first importance is that it is covering up the sugars that we need to break down, that we need to be eaten up by the microbes, right?
Okay.
So lignin is covering those sugars. The second importance of lignin is the structure of lignin. In the last 7 to 8 decades, several chemists and biochemists, they have tried to figure out the structure of lignin. And there are different schools of thought, on the structure of lignin, but most of the scientists kind of agree on that there is no one or definitive structure of lignin. There's no structure.
Oh.
It's just the way lignin is formed is that you have carbon to carbon bonds, or you have carbon oxygen carbon bonds, but those bonds are linked to each other in random fashion with about 22 different kinds of bonds. And I can say some of those bonds, those were beta, 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 alpha, beta 05, beta 07. But without going too much deep into the chemistry, again, I'll use my hands. If you have a carbon atom, if this is my one carbon atom, and this is my other carbon atom, if I'm attaching the carbon atoms together, this is one bond, right?
Okay.
But if I am attaching a phenol, which is OH, which is, the OH compound that we have in alcohols. If I'm attaching here an alcohol, that is another kind of bond. But what if there is no structure? I can attach a phenol here, but in the next segment or in the next carbon atom or molecule that I'm going to attach, the phenol attaches here instead of here, right?
Yeah.
So that's a different bond now. And then in the next ring, the carbon or the oxygen attaches at a different site, and then they attach at a different site. So the basic consensus about lignin is that it's a macromolecule with no structured attachments of all these different bonds.
Okay.
So there is no structure. So because there is no structure, it's really difficult to break it. Because now I don't know that am I breaking a COC, like carbon oxygen carbon bond, or am I breaking a carbon phenol bond, or am I breaking a carbon carbon bond? And if I am, where am I breaking that bond? Is that carbon attached to a fifth carbon, or a fourth carbon, or a oxygen attached to a second carbon? So the structure is so complex that even in nature, lignin breakdown is often the rate limiting step. If you go and study and read some of the publications in forestry, when a tree breaks down and when the scientists have figured out how the breakdown happens, lignin is one of the rate limiting steps, because even for the microbes living in the soil, it's difficult to break lignin because of the complexity of the structure.
So it's the structure. I mean, I'm sure the actual components of lignin play a role as well, but it's the structure that is really resulting in its resistance to decay by microorganisms more than most other.
Yes, sir.
Okay.
And there is another layer to the structure. So in lignin, there are phenolic compounds, and then there are non-phenolic compounds. Phenolic compounds is a carbon ring with a OH, with an alcohol attached to it, right? So it's easier to break down the phenolic components, the components that has an alcohol attached to it. It's easier to break them down. But it's difficult to break down the non-phenolic components of lignin. And that's because of the redox potential, the oxygen, the redox potential that's needed to break down. So we need a higher redox potential of anything that's trying to break down lignin. We need a higher redox potential to break down non-phenolic compounds.
And that's why lacase has that. It's a very high redox potential. I guess that makes sense now. Okay.
Yes. And lacase, again, there are different sources of lacase. Plants themselves, they have lacase enzyme. They produce lacase enzyme within the cells to help produce lignin.
Okay.
But that lacase has a very low redox potential.
Okay.
Now, the lacase that we get from fungal species, not all fungal species, but some of them have very high redox potential.
Okay.
So the higher the redox potential, it means that enzyme has the capacity or the capability to break down different components of lignin, phenolic as well as non-phenolic components.
Okay, so let me bring this to a practical world here a little bit, because you just said something I think that I on my channel I try to emphasize, is that just because this particular project found this result, it doesn't necessarily mean that this particular product that contains the active ingredient would also result in that. What I mean by that is lykase is not necessarily lykase is lykase. There's where the source of that lykase would likely have a different impact on whatever desired, in this case, thatch reduction. So if a product says, well, there's very few products that have lykase in them in the turf management. I don't know if maybe one, but it doesn't necessarily mean that because it contains lykase, it will likely result in that. We need to have a little bit more information of what organism it came from. That's going to complicate things. Right. That's going to complicate things. Can you imagine though? Because in the manufacturing and distribution, I doubt they're going to say this lykase came from this organism. Hopefully now, if somebody hears this, they'll include that. You know, if there's organism specific lykase that is productive and other lykase that is less productive, I think the consumer would need to know that. It would be nice if they did.
Yes.
You know.
And this is absolutely necessary. And the source of lykase that I used for all these trials, not all the trials that are in this paper, but for the thatch management trials primarily, we used lykase enzyme initially from Sigma Aldrich company. We bought it from Sigma Aldrich, and the source was the fungal source was Tremitis versicolor. Okay. And then later we also tried. And we used some lykase enzyme, and we tested different sources of lykase, and we tested some of the lykase enzyme that one of our collaborators in China, in one of the universities in China, was fermenting and producing lykase. So we also tested that lykase alongside the lykase that we were the lykase from Tremitis versicolor. Okay. So and sometimes, you know, we figured out that that the other source worked pretty good, and sometimes it would not, and that's based on the redox potential, the difference in the redox potential.
Okay, perfect. Well, that's enough for me, honestly. I'm gonna have a good day the rest of the day because I had my interest and my ignorance in this topic. I feel like I've gained something already today just with what we've gone over, and we have more to go over. So some of these, I'll give you just a brief background, Sudeep. Some of these graphs that you have reproduced from previous publications, my audience has seen, but people who are watching this for the first time might not have. So I wanted to kind of go through the papers, if you're okay with this, simply by using the figures, most of the figures, because you have several figures in here, and they really tell the story from my perspective, and I think the audience might be able to see what I'm talking about when we get to it. So this paper, or this figure here, for those people listening, I'm looking at figure 1 in this paper. If I can get it on the screen, if I can, I'll back out of here. And this paper, this figure was one we already went over with my audience prior, Sudeep. But if you want to just kind of briefly go, I believe this was the greenhouse study with creeping bentgrass, if I'm not mistaken. And
yes, this was the first study that we had, and see, we at the beginning, we were at a point where we understood theoretically, we understood the concept, right? Okay, yeah. And then we went to some of the lab studies, and we did a little bit of lab studies to figure out if lykase is actually reducing the lignin content. You know, we did some petri dish studies, and after that we knew that the concept works. But then we had no idea in terms of at what rate or activity level should we use lykase, what frequency should we apply lykase to turf grass, is there any impact on the turf quality? There were so many unknowns, and we had no answer. So if you look at this table, you'll see that at the bottom axis, you see that our lykase was all the way from 0.2 to 2.
Well, you actually had 20 as well. It's not on this particular graph, but you all went up 20 because you don't know the rate it would work. You don't even know if it will work, you know? So you just, like Paul said last night, you're just a shotgun approach, and we're hoping we find a rate in here that we do see a response. Imagine if you, Sudeep, imagine if you use 20, but the critical limit or the critical concentration for a response would have been 40 or 50 or 60. You know, you don't know. You're trying your best to get it on within the range, and you guys did. You picked some ranges, and you found that 2 units per square centimeter was the unit that resulted in a response. So for those listening, I'm sorry, go ahead.
And one more thing before, sorry to interrupt you. The G here is glycol. Now, as we were talking about redox potential, right? The capacity or capability of an enzyme to break down structures. Sometimes what happens is that in an enzymatic reaction, enzyme itself might not have a high redox potential, but in presence of a mediator, the potential increases.
Okay.
So because since this was our first study, so we wanted to test a mediator as well.
Okay.
So we tested guaiacol. But what we figured out was that lipase in presence or without the presence of guaiacol was working.
Yeah, you see it here. So we're looking at a figure that has thatch thickness and matte layer thickness, and matte layer thickness and thatch layer thickness.
Yes,
and we have a control, we have nothing, and then we have 0.2 units and 2 units with and without glycol. And basically what you all found was that nothing really occurred at the 0.2 concentration, but when you guys got to 2, you started to see the well, not the matte layer thickness, but the thatch layer thickness decline as a result of this inclusion of 2 units of lipase with or without glycol. So that was where you're like, okay, there's something going on here. And my audience, I think, is starting to become familiar with how our minds work in science. And that is, things are really expensive. We can't just go out in the field because the field research is the most expensive research. So we generally will start in the lab or in the greenhouse when we have exploratory research. And that's what that was. So now you guys knew around 2 was probably, at least we know something probably going to happen at that rate. And you started going out and doing more work. And let's see. Oh, yeah. Well, this graph right here is what we're going to talk about in a minute. But the graph on figure 2, and remember, there's some people who aren't watching this. There's going to be some people listening to this. We're looking at a similar graph where we're looking at enzymatic treatments, the 0, and then we have the 0.2 units, and then we have the 2 units, just like the previous graph. And in this case, we're looking at the soil depth from the top 1 inch or the 1 to 2 inch depths. And on the y-axis, we have, what is this on the y-axis? Total organic carbon, I guess. Total organic carbon.
This is organic carbon. Yes.
Yeah, and in the top inch, go ahead, Sudeep.
What I wanted to say here, that when we did our first study, we knew that the 0.2 unit works, right? But the first study, we were not able to see how effective is lipase in breaking down organic matter, and this is why, because in a live turfgrass system, lignin is constantly being produced by the plants, right? So we were trying to break down lignin while plant was producing lignin, right?
Yes.
So I hope I'm making sense, right?
Yes.
So the next step that we wanted to do was that now we know that 0.2 unit works. When we apply it every 15 days or every bi-weekly application.
You mean the 2.0 units? Is that what you mean?
Yeah, the 2.0 unit works.
Okay.
We knew that it works, but the first study didn't tell us the actual effectiveness of lipase in breaking down organic matter, right? So the second greenhouse study was done we killed our bent grass. We killed it, we clipped it all the way to the thatch. And then we started applying the same treatments as we did in our prior experiment. And we just wanted to see the overall impact of lipase enzyme on organic matter breakdown without plants actively producing organic matter.
Exactly. Okay. Yeah. And we saw that you all saw on the top inch that, again, this nothing happened between the control and the 0.2. But then you start to see a reduction when you increase the concentration to 2 units per square centimeter. That's when you see this reduction here in the top 1 inch of the thatch, I suppose. And then, so that's basically you're trying to, and I mentioned this before, and there's so many things going on. We try to limit everything as best we can and control everything just to refine, answer the question we're having. And when you have, like you mentioned, we're trying to see if there's an influence. Well, in the meantime, it's not a static system. There's still lignin being produced by the plant. So he's like, wait, let's just kill it, stop there, and see if there's anything going on just from 0. And that's what you found there. Now, when we go to figure 3, now this is what I think we talked about last night with Paul briefly. I may be wrong on this, but this is a very similar graph where we're talking about We have the enzymatic treatments from the control with nothing all the way up to 0, and then 2 units of lignin, and all the way up to 20 units. I'm sorry, lignin, 20 units of lipase. And you want to tell us about this particular graph? Do you remember you see this here? Okay.
Yes. This graph is also this is from the same study. I'm just trying to make sure that I do not misinterpret any graph. Yes, so this is from the same dead grass study, and this is the thatch layer thickness.
Okay.
So as we were talking about in the first graph, that yes, we went all the way to 20 units per centimeter square. So in the second study, again, we kept the treatments, and we wanted to see the impact all the way up to 20. What we saw, and if you can see here, is that after about 2 months, we started seeing the decline in thatch layer for all the way from 2 units to 20 units. But when we applied it for 6 months, it basically, we are just looking at the enzyme treatment. It was all statistically similar, except for the 20 plus glycol, which was different than at least one treatment. So basically, for us looking at this graph, it just the information that we got was that 2 units without glycol is basically doing the job.
Yeah. And it goes from, not doing anything after 6 months. The thatch was about 10 millimeters down to 6 millimeters. So you're looking at not only a statistical reduction, but a biologically significant reduction. We're talking about 30 to 40% reduction in thatch thickness just by the application of 2 units. So let's go down to the next one. And this is where it gets a little more confusing for me. Hopefully you can help me out here. I don't know if I can get it all on the page. Let me zoom out here. I think I'll have to zoom out on this so the audience can see the whole thing. But figure 4 was we're talking about glucose, and I can't even pronounce some of these words. Go ahead.
Yeah, these are the sugars. And see, remember, when we were discussing about the importance of lignin, and I said that we are actually looking at cellulose, a strand of cellulose and hemicellulosic sugars, and they are bound by lignin. So we are trying to open up lignin molecules so that microbes can basically eat up or decompose the sugars that are present in the cell wall.
Okay.
So when we did this dead grass study, that's where we saw that yes, lignins reduced, all the things that we were looking for. Thatch layer was reduced, organic matter was reduced, but the philosophy, that we had at the beginning of this project was that we will assist by applying lipase enzyme. We will assist in opening up the structure of lignin, and then that will second-handedly assist the microbes to reach the cell and decompose different sugars. So that's when I tested, on a weight basis, I tested the different sugars, monomers, because cellulose and hemicellulosic, these components are composed of different sugar monomers. So they are composed of glucose, arabinose, and so we just tested some of them and also tested total sugars. And as you can see, as we increased, as we moved from 0 or control to 2 units, you at about 6 months, you'll start seeing reduction in sugar components, different individual sugar monomers. And that was basically aligned with our initial thought process that's what's happening.
So it's confirming to you that what your hypothesis was is probably what's going on, because it's opening it up. You're seeing not only the reduction in thatch and lignin, but you're seeing reductions in the sugars that theoretically should have been reduced, and in fact they were. So this is the paper, or figure 5 now. Remember, this is the paper we talked about last night with Paul, where we're talking about Bermuda. So now you did those greenhouse studies. There's clearly sufficient evidence to indicate there's probably something going on here, but it was in the greenhouse, and we can talk about all the differences in greenhouses and field. There's clearly importance, and what we find in the greenhouse doesn't always translate in the field, but all of our clients, with very few exceptions, are working in the field. So now you take it out to the field and do some Bermuda grass and zoysia grass work, and we find that here in this figure. You want to go over this?
Yes, sir. So by the end of the first 2 greenhouse studies, we knew that if we apply 2 units of lipase every 15 days or every 2 weeks, once every 2 weeks, for about 6 months, we see the reduction. We were able to see thatch reduction, organic carbon reduction. We were able to see reduction in the sugar. So we were pretty confident that in a controlled system, we know what works. But now let's try, and we also knew that it works in creeping bent grass. So we knew that, okay, now let's move out in the field and also try different turf species. So we tested it in Bermuda grass and zoysia grass. The baseline that I have for both these grasses was the thatch layer or organic matter for this graph, organic matter at the beginning of the study.
Yeah.
And so if you see in the control, plot where we did nothing, you see an increase in organic matter over time.
Over the baseline.
But over the baseline. But with our treated plots, we saw a reduction. In our organic matter. Yeah.
And this is what I told Paul last night, is that when your Hort Science paper came out in 2012, I think it was, the greenhouse study, I read that, and I told Paul this last night. I was like, I read that and go, okay, but I've seen something like this before. I've seen blackstrap molasses do something. I've seen inoculated fungi do something here and there, and then later on it doesn't work, and it's hit and miss. There's inconsistency in the results. So that's when I read the Hort Science paper. I'm like, okay, let's check that and put that in the mental bank of potential interest. Then this paper comes out, and I go, man, Bermuda grass, I can buy that. I'm like, okay, Bermuda grass, okay, there's something going on. But then when you see it with zoysia grass, now granted, you didn't see it in the second year here, but but the first year, I'm like, okay, if you're gonna show data that shows a reduction in zoysia grass thatch from a bottle, essentially, if you want to, kind of using that phrase, then you got my interest. I'm like, okay, there's something to this, and this is what really, started to move me off the center line of neutrality. I'm like, okay, there's something probably here. I gotta look at this. So this, I just want to let you know, as a scientist, this is the paper that started to convince me. The Hort Science paper was just like a little thought to me. And then this paper is like, okay, all right, I got to look at this. This is something that could be a game changer, really. And so good job on that one. So now we're going to go to figure 6, which we're building here, guys. Remember, this is epistemology. One paper isn't going to really move me off the center. We're not talking about one paper. We're talking about 2 or 3 or 4 or 5 papers here. And this is the next part here is figure 6. I'm going to leave this on the screen because I think it's a little too big to fit in when I zoom in. This one is cultural management practice. We have a control. We have cultural management practice. I don't know if that was an aerification or verticutting. It just labeled as cultural management practice. And then you have the applications of lipase on the x-axis. And then you have on the other panel, you have the frequency of lipase applications. And on the top panel, you have the rate of lipase applications. And in all cases, we're looking at millimeters of depth of thatch layer thickness on thatch layer thickness. Okay, walk us through these 3 panels here.
So this to me, I would say, was my second best paper, or second best result, because the best is the last one. Okay, and this is the second best because after all these studies that we did and we shared up till now, right? We kind of were testing to figure out if we know the range and we know how frequently we need to apply, right? So after that 3 papers, we were pretty confident that 2 units of lipase applied every 15 days for a period of 6 months, yes, it will work on thatch, right? Okay, yeah. Now the next logical question was, can we further improve the efficiency or reduce the applications of lipase, right?
How low can you go? Yeah.
How low can we go, right? And in all fairness to everybody here, lipase enzyme, the way, as I mentioned, that we were purchasing it from Sigma Aldrich, and this was not something that was very cheap. It was expensive enzyme to test. So Dr. Raymer and I, when we all sat down, Dr. Carrow, Dr. Raymer and I, And it was just a unanimous decision that there is no way that we can do a complete factorial design and we can test what's the lowest frequency and what's the lowest rate that we can go. So we divided our rates or our treatments into blocks.
Okay.
And the first block, the top block that you see in this paper, is we were testing how low we can go on a rate.
Yeah.
So we know that the 2 rate works. So we chose to go as low as 0.5,
right here,
then 1, and then 2,
yeah.
Yes, and then a 1 rate above 2, which was 4.
4, yeah.
And in the parentheses, you have frequency of application, which is every 2 weeks.
Okay.
That's why you have the number 2. So now looking at that top panel, what we knew now after this study was that we can reduce our rate from 2 to 0.5.
Yeah. So
and
you just reduce the cost by 75% or whatever by 25. You're applying only a quarter of what you were applying.
Only a quarter of what we
and
were applying.
You're seeing the same response.
We were seeing the same response after 6 months.
Okay.
All right. Now the middle panel is where we kept our rate the same rate, what we know works, which is a 2 rate, 2 units of lipase. But we changed our frequency from 2, applying once in 2 weeks, to once in 4 weeks, to once in 8 weeks, to once in 12 weeks.
Okay.
All right. So what we saw here in the middle panel is that we can, if we are choosing 2 as our rate, we can go as low or apply lipase as low as once in 4 weeks.
Yeah.
And we'll get the same result. When we use the 2 units, but go to once in 8 weeks and once in 12 weeks application, we start seeing a little bit of buildup of that, which was,
Yep, I see that. Yeah.
So from this, from the rate and the frequency study, we knew that it's wiser for us or for anybody to go low on the rate, but keep the frequency once in every 2 weeks for better results.
Okay. Yep. And then the bottom panel, you included it with some culture management, and you see the same response.
Yeah.
This is, to me, Sudeep, this is, I don't know how this sounds to you or not, but this is more practical applied. Like, what is going to happen relative to what we're already doing, right? Cultural management,
yes, sir.
Right? And
yes, sir.
Go ahead and explain.
So our cultural management was co-aeration twice and filling the holes with the scent-off dressing. So that was our cultural management practice. And we wanted to, and in this set of treatments, we wanted to compare the effectiveness of lipase alone with the cultural management practice and along with the cultural management practice.
Yes.
So one, and this is one of my, that's what I said, the second best result that I was very happy was with the cultural management practice. When you apply lipase at 2 units once in a month with your cultural management practice, that was the best result that we got.
You have to understand, and for those listening that aren't necessarily scientists, we don't know what's going to happen. We do all this work, set up all this study, hopefully have some funding for it, scrape together the resources we need to do it. We go out and we do it, and it takes a year, 2 years, whatever it is. And only at the end do you start to see the results. And so when these results come in, Sudeep, how happy were you?
Very, very happy.
I mean, it's like, wow. I mean, this is actually working. Imagine, and it does happen where you do all this work and nothing happens. That happens a lot. But in this case, we see the lipase applied at 2 units had a similar result as the cultural management, but combined together, they had a more significant impact when they were used in conjunction with each other. Very, very well done. I like that figure a lot. Let's go to figure 7. It's a line graph. I don't know how much you want to go into that. It's a pretty simple little line graph.
Yeah. But this is my best result. This is the one that I love.
Okay. Let's hear it.
So now with the last study that we discussed, right, we were at a point where we knew that we can reduce our lipase rate or application dosage from 2 units to 0.5 and keep on applying it every 2 weeks for 6 months, and that works, right?
Okay.
Or we reduce the frequency, it still works. But the next question was, and this is very obvious question. Now I want you all to think about it. we are just opening up lignin bonds, cellulose. Remember the cellulose that we have and the lignin that's on top, right? So we were just opening up the lignin structure so that the microbes are able to decompose sugars. When the sugars are gone, right, what's left is whatever lignin is left is your primary now component of your organic matter, right? So if we keep on applying lipase and we keep up opening lignin structure, and the bacteria or the fungus in the soil, they're able to keep eating whatever sugars are available, there has to be a plateau. When you reach that, when you hit that mark where now what you have in your organic matter is primarily lignin.
Yeah.
Right. So that thought led us to think about to have this study where we wanted to see, okay, what's the residual impact of it, right? If I have to develop a protocol on a yearly basis to manage thatch with use of this lipase enzyme, till now all my research was apply it for 6 months, see the results, apply it for 6 months, see the results, right? And we knew we are getting the results. But the question was, what happens after those 6 months?
Yeah.
I applied it for 6 months and then I stopped applying. What happens? How much time it takes for thatch to start rebuilding? And then the next question was, when thatch starts to rebuild, what do we need to do next to bring it back down, right?
Yeah.
For example, to make it a protocol, like what should be a protocol on a yearly basis to manage thatch using lipase enzyme? And that's what the gist of that study is in this line graph.
Okay.
So I'll try my best to explain it to everybody.
Yeah.
So the 0 here is the starting point of the treatment. That's when we started applying lipase enzyme.
Okay.
So the first three bars that you see are for our control. The middle bar is for lipase enzyme that we got from Sigma Aldrich, and we applied it at the same activity level that we know that works, which is 2 units applied at every 2 weeks. And then we were also testing the lipase enzyme that we got from one of our collaborators in Nanjing University in China. And that's the CHU because it's Chinese university lipase. So we labeled it as CHU. So that's the lighter color bar. So and the length of that bars that you see from 0 to 6 is that shows you the treatment, the zone where we applied the treatment.
Okay. Yes. So you just applied it here and then you had a gap and then you applied it again. Okay, I'm with you.
Yes. So when we applied the treatment, if you can see, both the Sigma Aldrich enzyme and the Chinese university enzyme performed equally well, and our thatch layer decreased, the thatch thickness decreased.
Yes.
Then we stopped at 6 months was our end period for the treatment application. So we stopped there, and then I came back at the 12 month after a gap of 6 months, and I measured thatch layer again. And as you can see, that's the residual impact of lipase because there was no thatch buildup.
So it's maintaining that effect for at least 6 months after application stop.
After application, yes. Now, I came back after 18 months and I tested thatch layer on the middle line. Don't look at the bottom line, but look at the middle line.
Okay.
This is where I did not apply. This was the lipase enzyme from Sigma Aldrich. So these are the treatments where I did not apply lipase for a second 6 month period. And you see that there's a buildup, starting up of thatch buildup there. But in the Chinese university treatment, I applied a second set of treatments from 12 months to 18 months, which is a second 6 month period after a gap of 6 months. And you see that the thatch layer there wasn't any buildup of thatch.
Stayed the same, I don't know which one I'm more impressed with, this line or this line, because here, this darker middle line, it hadn't had any application of lipase in a year, and it was still less than what would have occurred had you done nothing. It was still showing a reduction here. So,
yes, sir.
Brilliant. Brilliant. A brilliant idea to run that one. That's nice. Let's go to the next one.
So this.
Go ahead. Go ahead.
Well, I was about to say that this was the last study of my PhD program.
Okay.
And as you were mentioning, when you get results, was I happy or not? Yes, because we started from the theoretical knowledge of lipase and theoretical knowledge of lignin, and we had no clue what rate, what frequency would work and what would not work.
Yeah.
So, but at the end of my PhD program, I was able to kind of say that, hey, if we have this activity of enzyme and we apply it at this frequency, we need to apply it for 6 months in a year. And we have to repeat that the next year if we want to maintain our thatch.
Yeah. So you're starting to, I mean, in some ways, I'm seeing this as a means to figure out how to make it financially feasible for end users. You don't have to necessarily apply it all year long. You can do a cycle, I guess if you want to call it that, and then take a break and do another cycle. And you can still see or realize some benefit to that in terms of thatch reduction. So, when you're dealing with a management practice like this that is fairly expensive, we got to figure out how to make it fit financially and for the people making it to make money and so forth. So trying to refine it down, and this is a perfect example of what I've talked about on my channel about doing calibration studies. We're picking rates. It doesn't mean the high rate you should use. We're just trying to pick out rates, and then we can refine it down once we see a response to the best management practice rate, right? Whether it's nitrogen or phosphorus or whatever the case might be. And that's what you're doing here with lipase, the exact same process. So very well done. Let's go to, let's see. Now basically all the next section, Sudeep, is soil water repellency. Do you want to take a break on the stop right here, and then we can maybe address soil water? Why don't we do that? Why don't we take a break with this? And maybe if you're so inclined and you feel comfortable, you can come back, or maybe I can get Paul back and we can talk about lipase as a product for soil water repellency. That'd fit better, I think, when I go over wetting agents and so forth. Is that okay with you?
Yes, sir.
Okay. Because I think the rest of this, I'm not mistaken. Yeah, this is percent water volumetric content. Yeah, all the rest of this is the soil water repellency stuff. So I've learned so much here. It's going to take me, a week to digest all the information. But that's the reason I got you on here, because I read that second or third section about the lignin structure and the lacase structure and the functionality, and I just like, man, I don't understand this. And you did a very good job educating me on exactly what it is that the lignin, what the problem is with lignin, right?
Yes. Well, one thing I think we did not address in our podcast today is why we chose lipase.
Okay, go ahead.
Right. I guess this is very important, because there are other enzymes that do the same thing, that can break down lignin. Yes. And maybe they can break down lignin better than lipase can. But the question is why we chose lipase. There are other enzymes such as lignin peroxidase, manganese peroxidase, versatile peroxidases. All three of them have similar functionality as lipase, and they can break down lignin phenolic and non-phenolic bonds. But all three of those enzyme classes, they need a strong oxidant, such as hydrogen peroxide, to initiate their reaction, right?
Okay.
And so those were just, there wasn't any thought of using them on turfgrass because they need a strong oxidant to initiate their reaction. Whereas lipase enzyme, on the other hand, needs oxygen.
Okay.
Which is readily available. Which
yeah,
is in our
is.
air. So it just needs oxygen to carry out its reaction. So that's why we chose lipase.
That's a very good explanation. I didn't realize that. And I see you wrote that in the introduction here of this paper. If anybody's interested, you can actually, I believe this paper is open access. I think isn't the International Turfgrass open access now? So anybody can go download this. I think you can actually search for this on Google and it'll pop right up. Otherwise, go to TGIF, the Turfgrass information file, and you can find this article and download it. And what Dr. Sidhu is saying is noted in the last paragraph of the introduction that I apologize we didn't go over, but I appreciate you mentioning that because I was unfamiliar. I do know, unfortunately, I hesitate to even say this, there are some people who actually use hydrogen peroxide. I strongly encourage to reconsider that when you're dealing with an oxidizing agent that is potentially as dangerous as hydrogen peroxide when you're going out and putting it out in turfgrass systems in agricultural settings. You got to be extremely careful with these oxidizing agents like hydrogen peroxide. So that's why you didn't pick those other enzymes. You can just pick lipase, and it uses oxygen as it is. So very well done. Wow. Okay, Sudeep, I'm going to have to decompress a little bit here. Okay. But like I said, I understood what was going on in terms of, okay, I'm seeing these results. I'm still not entirely clear, but I wasn't real comfortable understanding what exactly is it about lignin and what exactly is it about lipase that's doing this. Why is it a problem? So thank you so much for coming on and helping educate myself and my audience as to, what's going on here with these.
My pleasure, and I enjoyed our conversation.
Good. Well, I'm glad you did. Well, you're always welcome whenever you want to go. Like I said, I'll probably at some point get into wetting agents, and maybe I'll have you come back in and go over the last part of this paper, because there's a couple different ways to look at wetting agents. In this particular product, we're looking at removing the hydrophobic organic coatings. I'm assuming, I haven't read that paper, but you're actually attacking the cause of the problem rather than the symptom of the problem. I would assume that's probably the angle you guys were taking. I haven't read that paper, but interesting approach to that. Thank you so much. I really appreciate it. For the rest of you all, I will be back on next Monday at 10 a.m. As far as I can tell, next week will be a normal week, 10 on Monday, Tuesday, Thursday, and then 9 p.m. on Wednesday. And I'm going to do everything within my power to finish thatch next week and then move on to another topic. Because I was telling Sudeep, my audience is like, I have one little lane of turfgrass soils and the system that I stay in. And every time I go out of my lane, which thatch is definitely out of my lane, I'm not comfortable. I end up, getting ran over by the car going by me who doesn't know what he's doing. And thatch, I'm just not real comfortable with. And I really appreciate you educating myself and my audience today on this topic. So thank you so much. With that, guys, thank you, Sudeep. Hang on tight, Sudeep. I'm going to close this off. I'll be back to you. For everybody else, have a great week and a weekend. And I'll see everybody on Monday morning at 10 a.m. Thanks. Bye.
Thank you.